Good afternoon and welcome to Uni4Me Live. I'm delighted this afternoon to be joined by Dr. Sam Jones from Staffordshire University, and later on Professor Kate Harvey will be joining us as well from Reading University. But initially I'm going to hand over to Sam, who's going to be speaking to us all about biological psychology.
We get the screen shared. Can you see that?
That's coming through loud and clear.
Lovely stuff. Okay. Hello. I am Sam Jones, Dr. Samuel Jones, and I am a biological psychologist or a cognitive neuroscientist or a neuropsychologist. I'm never really sure which. I'm one of those. And I'm going to be talking today about some myths about biological psychology. So 3 myths. Just before I get started, a little bit of a content warning. There is medical images and detail and stuff in this talk. So if you're a bit funny about that kind of thing, you might want to look away on those parts, but not too much. Just a warning there. Okay, so whenever I'm talking to people about biological psychology, I like to sort of set out, first of all, an answer to the question of why psychologists care. Why do we care about cells and biological things? Because, you know, psychologists care about the mind, right? They care about how people think and why people behave certain ways. So why are we interested in cells and gooey stuff like that? Well, my answer to that question is that every thought that we ever have happens physically in our brains. And I think this is an amazing idea, that everything that we think about, all of the things, the fact that I'm talking to you now, and hopefully you're listening to some degree, means that I'm making something happen in your head, right? There's some sort of physical thing, electrical thing, chemical thing happening in your brain that lets you think certain things and lets you behave a certain way. I always use this little example. So here's me sitting, supposed to be probably marking essays, and instead I'm daydreaming about cheesecake, which I do probably too often. Love a bit of cheesecake. So if you could zoom in and look inside my head, and that's an MRI scan of my actual brain, isn't it lovely? Very proud. And then zoom in further, and I've gone for somewhere near the back here because I'm visualizing something, and there's lots of sort of visual areas towards the back of the head. If you could zoom right in, while I was thinking about cheesecake, you'd see that there are specific things happening, specific neurons firing that are letting me think about cheesecake, that are sort of making this process happen. And that's you know, like I said, I think it's a really sort of amazing thing that everything that we think, everything that we do is because there's something actually happening in our heads. And that's why psychologists care about biology, because it's biology that makes all of the psychology stuff happen. I will, of course, point out that our brains work together with the rest of our bodies. So it's not, you know, you're not a brain in a jar, right? You've got a brain and it's connected to the rest of your body. And it's all of these things together that lead to sort of our ability to behave and do stuff in the world, to think. So one example, you know, is that our senses are telling the brain about the world, right? We can see what's going on, we can hear what's going on. A little aside, because I always think this is quite a nice thing to think about, is the question of how many senses have we got? How many senses are there? And of course, there's, you know, the classic answer is there's 5, right? So we can see, there's sight, we can hear, there's hearing, taste, smell, and touch, right? Those are the ones that you're always taught about in school textbooks and things, and then, you know, they're the 5 senses. I like to do a very little demonstration to sort of prove that there are more than 5 senses. So anybody who's watching this right now or a recording, if you just close your eyes for a second and you touch your nose with your finger a few times, hopefully most of you managed that okay. And the question I would ask is, what sense did you use to do that, right? It wasn't sight, because hopefully if you did as you were told, you had your eyes closed. It wasn't touch because you weren't touching anything until you touched your nose, right? How did you get your finger from where it was to the end of your nose? And the answer to that question is that you used a sense that people don't talk about very often called proprioception. Proprioception is the sense of where our limbs are. And there are lots of other senses that we don't even think about when we're listing them. So balance, for example, there are little organs in our ears that tell us whether we're upright or sideways. Or you can sense temperature, when you're hungry. These are all different senses that you don't necessarily think about, but there are tons of them, not just the 5 that we talk about all the time. Anyway, that was a little bit of an aside. But moving on. So we've got our senses telling our brain about the world. Part of my research is actually interested in how our brain is able to combine lots of different senses to help us better understand what's going on in the world. But then, of course, the brain needs to tell your muscles to move, because a lot of the time that we're sort of thinking about stuff, the reason is because we want to do something with it, right? So you see a glass of water, your brain says, pick up the glass of water, and your muscles do the thing. So it's this constant communication between the brain and the body. And biopsychologists are interested in this kind of thing. As I say, electrical and chemical signals constantly talk back and to between the brain and the rest of the body. Okay, so that's what I would consider, myth number 1. The first of the 3 myths we're going to think about today is that this idea that biopsychology is all about brains, that people who are doing biopsychology or neuroscientists, cognitive neuroscientists, like I say, there's a lot of overlap between these various job titles, but we all do kind of similar stuff. Biopsychologists actually have lots of different tools, and they're not all focused on the brain. So, for example, there is something that we can measure called electrodermal activity. And what that is, it's basically measuring sweatiness. Bit of a gross image, but that's what it is. It's kind of, you measure how sweaty someone is very, very precisely. And what that does is it lets you see how emotionally somebody is reacting to something. So you play them a scary video or something, and you measure this electrodermal activity. You can see how kind of scared and stressed they're getting by the scary film. No brain measurements there. You're just measuring sweatiness of somebody's fingers. Biopsychologists also will do testing with drugs, right? And that could be sort of reasonably, everyday drugs like caffeine, like you get in your coffee, or it could be much more sort of complicated drugs. And you can see how people's thoughts and behaviors change when they have drugs versus when they don't. You can measure what we call biochemistry, so levels of different chemicals in the blood or in the different parts of the body. We can do genetic testing, so we can have a look to see whether a person's genes can change how they behave. No brains here so far. Animal testing, so you can test animal behavior and things like that. We can look at ECG, so measuring the heart and heartbeats and looking at things like heart rate or heart rate variability and how that is associated with different thoughts and behaviors and things. So again, another thing that biological psychologists do that are not focused on brains. And then of course there is all of the brain stuff as well. So we've got our EEG with the cap, and we've got fMRI. We've got functional near infrared spectroscopy in the top right over there, another different kind of funny hat. TMS. We've got all sorts of different things that are also focused on the brain as well. But yeah, I think that it's important to sort of bust this myth that if you're a biopsychologist and you're focused on the brain, you might because obviously a lot of the stuff that psychologists care about happens in the brain, but there's lots of other stuff that we do too with other parts of the body. Okay, now over to myth number 2, which is I think my favorite and the one that I'll maybe spend the most time on. So myth number 2 is the idea that as a biological psychologist or as a neuroscientist, I can read your mind. So as long as I've got the funky cap on you, you can see in the picture there, or as long as I can see inside your head because you're lying inside an MRI scanner, I can read your mind. So we can't do that. And to explain why we can't do that, I thought I'd go into a little bit of detail about how these 2 particular things actually work. So starting with EEG, and there's a picture of me a few years ago with an EEG cap on. You might have seen these things before, right? They are used sort of medically sometimes for diagnosing sleep problems and epilepsy and things. But yeah, it's a funky cap that you wear, and it helps us read the electrical signals in your brain. So that's what it's actually doing, is it's measuring your brain's electrical activity. So you probably know that when the brain's doing stuff, there are electrical signals firing. And what EEG is doing is measuring those. It has to be very, very sensitive, of course, because they're tiny little electrical signals, and they've got to get not just, you know, from the brain, they've got to go through the skull, through your scalp, and then be picked up by these things on the outside. So they have to be very, very sensitive little sensors on these caps. But that's what EEG is doing. So it's measuring the actual electrical activity of your brain, which is cool. I've got a video here, hopefully this will play okay, that one of the technicians in our psychology department at Staffordshire recorded, just showing you some of the equipment we use for EEG. So that's the cap itself. Here's some weird gooey gel. We've also got some wipes and alcohol for cleaning up and scrubbing, and I'll show you in a minute what some of that's used for. We've got some syringes. I'm going to fill those up with the gooey stuff. I'll show you that in a moment. Some pretty scary looking needles, but these aren't used for going under the skin. So they're not needles for poking into somebody. And then some extra long cotton bud type things. A tape measure, because we have different size caps for people who've got different sized heads. So a tape measure is good to see how big somebody's head is. So what we do is we'll take this needle, we'll stick it on the end of the syringe. Like I say, this is not actually going under somebody's skin, so don't worry too much. We take this really disgusting looking goo. It will go into the syringe, and then for each of these little electrodes, we will squirt some of this goo through the hole. And what that does is it lets us get a really nice connection between the electrode and the skin. And what's happening there is this technician is scrubbing a little bit to help move the hair out of the way and make it easier to connect the electrode to the skin. So what the gel does, and that little bit of scrubbing as well, is it basically means that we can get a really nice connection between the scalp and this electrode thing here, so we can pick up as much brain activity as possible. We have to do this for every single one of the electrodes on the cap, which can take a while. Some of these caps have 128 or even more of these little electrodes on, so it can take a little while, but it's worth it because you get a much nicer connection if you use the squirty gel and give it a bit of a scrub. Oh, not again. There we go. So what can we see with EEG? What are we actually measuring? There's a couple of different things that you might want to look at with EEG. One of these is what we would call oscillations, or you might have heard of as brain waves. Basically, we're measuring how, you know, as the brain's working, we get these sort of wobbles in the signal. And what EEG can do is measure those wobbles. So we've got them over here. And they can tell us all sorts of things, including about sort of consciousness, how awake somebody is, for example. They're, of course, named after Greek letters. So we've got these really, really fast wibbles that go up to 100 times a second. So we call these gamma waves. And then they get slower through beta, alpha, theta, and delta. Named after Greek letters because scientists love a good Greek letter whenever we can. And they can tell us things, like I said, about sort of consciousness, sleep, that sort of thing. So these delta waves at the bottom, for example, are really, really slow changes in brain activity. So we could say maybe 4 or maybe even sort of 2 wibbles per second, really, really slow. And you only really see this if a person's in quite a deep sleep. Whereas if you look a bit higher up at these alpha waves, these slightly faster wibbles, you can see these quite clearly on an EEG signal. If you get somebody to sort of sit still, close their eyes and relax for a few seconds, you start seeing a really nice clear alpha wave. So they're awake, but they're relaxed. That's where you get these things. So that's one thing we can measure with EEG is brain wibbles, oscillations, brain waves. And that can tell us about what stage of sleep somebody's in, for example. If they're asleep, we can see if they're in a lighter or deeper sleep based on these wibbles. Another thing we can look at is something called event-related potentials, or ERPs. And what this can tell us is more about sort of when the brain processes information. So say you get somebody looking at something on a screen, so a picture or a flashing light, or they listen to a sound or something, and you get them to do lots and lots of times, same thing. what you can do is you can look at the EEG signal and see kind of how quickly the brain is processing that information. So it tells you in terms of milliseconds, so like thousandths of a second, really sort of precise information about exactly when the brain is processing stuff. One of the most common examples is when looking at faces. We get these little wibbles here, and we see a sort of a big spike a little after 100 milliseconds, and then a big sort of down spike a bit closer to 200 milliseconds. And what these are showing is sort of different stages of processing faces. So it gives us really good information about when the brain processes information. What EEG can't do is tell us where in the brain something happens. It can tell you roughly. It can say something that was going on kind of at the back of the head-ish, or kind of over here-ish. It's very bad at saying actually where stuff's coming from. So it's very good at when, very good millisecond when, but not very good at where. And that's because by the time you've measured the electrical activity that's coming from the brain, and it's made it all the way through the skull and the scalp, and it's got to your little electrode, it's really spread out. We'd say it's smeared. And it's really hard to focus and work out exactly where in the brain that signal came from. It also can't look at any parts of the brain that are below the surface, so what we call the cortex, so the sort of the outer part of the brain. EEG, very good. Anything that's deeper inside the brain, EEG doesn't have a clue, can't see that at all. And so it can't read your mind. It can give you interesting looking wibbles and squiggles and spikes that are very useful for specific questions, but it certainly can't read your mind. Okay, so moving now on to MRI. So MRI, again, something that you might have seen at some point or heard about, and basically it's a giant, huge magnet. And it uses this huge magnet, which you lie inside, to see inside your brain. Or it can see other body parts as well. You know, say if you hurt your knee or something, you might get an MRI scan. It can see inside any part of the body. I'm interested in brains. So that's a picture of my fiance lying in a mock MRI scanner there. But yeah, basically a person lies inside a very noisy tube, and we measure things going on in the brain using a massive electromagnet. There's lots of different flavors of MRI. So one of them is structural MRI. So we can look at the shape of the brain. That's my brain there. That's the shape of my brain. So it lets you have a really detailed look at the exact shape of somebody's brain. Right? And you can see, for example, that this shape changes as people get older, just like people's sort of muscles and things get smaller as they go into old age. That happens with brains as well. They shrink a little bit. We can also look at something called functional MRI or fMRI. And this isn't looking at sort of the shape of the brain. This is looking at brain activity. What it actually measures is blood flow. It measures kind of which part of the brain the blood's going to. And we like to have a guess that sort of means that a person's using that area of the brain more. So if we see more blood flow on the right-hand side, then that might mean that while somebody's doing a particular thing, they're using that side of their brain a bit more. But it doesn't measure the electrical signals in the brain. It's just measuring blood flow, which, kind of sort of shows us where the activity is, but not super precisely. We can do cool, colorful things called diffusion tensor imaging. And this can show us where all the different kind of connections and pathways are all over the brain. That's very cool. Very noisy, that one, if you're lying in the MRI scanner, but very cool. Gives you pretty colorful pictures like this. And we can even do something called spectroscopy, where you can measure the level of different chemicals in different parts of the head as well. Things we call neurotransmitters. So measuring chemical concentrations in different parts of the brain. So again, what can't MRI do? It can't measure electrical signals in the brain. We're measuring blood flow, but we're not measuring what the brain's actually doing when it's sort of doing its thinking stuff. And it can't read your mind, just like EEG. And then I've put a spooky font saying, or can it? Because actually, we're getting a bit closer to being able to read your mind using MRI. And I'll tell you a little bit of an example of a way that we kind of sort of can read minds using MRI. So to do this, I'm going to tell you about something called vegetative state. And this is a thing where a person's in hospital and they're totally not responsive. So they look like they're awake. They'll have their eyes open generally. So they're not in a coma, but it's a lot like being in a coma because they don't seem to be aware of anything going on around them. They don't respond to anything. So it's a lot of being a coma, but they're not asleep, but they're not responding to anything. So a researcher called Adrian Owen and all of the people he was working with, of course, wanted to test whether some of these patients might actually be aware, but they can't respond. So they might be able to know what's going on around them, but they can't make any movements to let people know that they know what's going on around them, it's very hard to tell, right? Because if a person can't respond in any way, how do you know if they're actually aware of what's going on? So these guys thought maybe we can use MRI for this. So they put the patients in an MRI scanner and they gave them a really simple instruction. They said, either imagine playing tennis or imagine walking around your house. So the person in the MRI scan doesn't have to do anything physically, they just have to think about different things. So healthy brains look like this when they're given these instructions. So if you say, imagine playing tennis, the top picture is kind of what happens. You get these kind of motor activations, movement areas, because a person's thinking about moving around and stuff. Whereas if you're imagining walking around your house, instead you get these activations as more like sort of memory visualization. So the way the brain works in these different situations looks very different. And so you can tell which of these two things a person's thinking about when they're in the MRI scanner. And what they did was they did the same thing with patients who were in a vegetative state, who weren't able to respond. And this is what they found in some of those patients. They found that when they gave them these instructions, they saw some brain activations that actually looked the same as they did in healthy patients. And you can see from some of these pictures that some of these patients had really, really bad damage to their brains, right? This first person at the top left is missing a big chunk of their skull there. So what they showed with this is that actually some of these people are aware of what's going on, but they've got no way of responding. But they can think about certain things when they're told to. So it showed that these vegetative state patients are not all unaware, they just can't respond to anything. So some could even use this to answer questions. So they could say things like, is your father's name Alexander? If yes, think of tennis. If no, think of walking around the house, right? This person, you can see they were thinking about tennis, the tennis part of the brain lit up. And yes, that person's name, father's name was Alexander. So it's pretty cool. That's sort of a little bit like mind reading. And a person's thinking about one of two things, and you can tell which one of those things they're thinking about by them being in an MRI scanner. We're getting much closer every day to sort of mind reading and being able to tell what somebody's thinking about when they're in an MRI scanner, but we are a long way away from it at the moment. We can tell between different things that you might be thinking about. And we can't, if you are lying in an MRI scanner and you suddenly start thinking about Pokemon or something, the person running the MRI scanner wouldn't be able to know that yet. Maybe in 10 or 20 years, we might be a bit closer. But at the moment, none of our tools let us read your mind. And the last thing I wanted to talk about very quickly, I'll just put some pictures of some reasonably boring looking research papers, but this is kind of the evidence for this. This idea that biopsychology, so neuroscience stuff with brains, is somehow more sciencey than other kinds of psychology. And there's a lot of research showing that if you make some made up claims, but you throw in some neuroscience-y sounding words, or you put a picture of a brain next to it, people are much more likely to believe and trust the information. And it's because I think you do get this sense that if people have got pictures of brains and stuff, it's all very cool and sciencey, and you trust it. Whereas other kinds of psychology can sometimes feel a little bit more kind of wishy-washy. Right? Well, actually, biopsychology is in a lot of ways no more scientific, sciencey than the other kinds of psychology. There's lots of ways of measuring how people behave and think that doesn't involve strapping any kind of wires to them or putting them in a noisy tube or anything, right? We can just ask people questions or get them to sit down in front of a computer. And we use very, very scientific methods to do that kind of stuff. So even though it sometimes looks a bit more sciencey, you know, because there's some pretty colorful blobs and wibbly lines next to the words, actually, there's nothing more scientific about biological psychology than all of the other kinds of psychology that you might be interested in. And I say that as a biological psychologist, so I'm biased and I still don't reckon it's more sciencey. So just a quick recap, the three things that I've talked about today, the three myths. So first of all, it's not all about brains. Biopsychologists can strap wires to all sorts of different parts of your body, and we can do all sorts of different things that are not just brains. Biopsychologists and neuroscientists can't read your mind yet, but maybe one day. And biological psychology or biopsychology, neuroscience, is not more sciencey than other types of psychology. It just looks it because the pictures are a bit prettier. That's it from me. Thank you very much for listening. If you want to hear more from our department, so Staffordshire Psychology, that's the Twitter handle for Staffordshire University Psychology Department, Staff Psych. Or if for some reason you wanted to follow me on Twitter, that's my little Twitter handle there. I don't tweet that much, to be honest, so feel free not to. But that's me done anyway. Thank you very much.
Thank you, Sam. That was absolutely fascinating. So I think now we're going to move on to our next contribution. That's from Professor Kate Harvey from Reading University, who hopefully will be joining us a little bit later on. But I should be able to share her screen, and she's going to be talking about research methods.
Hi there, my name's Kate Harvey and I'm a Professor of Health Psychology at the University of Reading, and I'm going to talk a little bit in this presentation about qualitative research methods in psychology. So looking at the psychology A-level, I've tried to identify some of the things that you cover in your A-level. And I've got a list here, and these are things that I will be mentioning in this presentation, so you can kind of try and match the two things together. So research methodologies, we tend to think in psychology about research methods falling into either quantitative or qualitative. And as I'm sure you've probably learnt in your A-levels, quantitative methods tend to focus on numbers, whilst qualitative methods tend to focus on words. When we conduct quantitative studies, the aim is to measure, whereas with qualitative studies, we're thinking more about understanding or explaining. With quantitative methods, you would always have a hypothesis that you're testing, whereas qualitative is more exploratory, so tends to be there's been an observation and you have a question, but you wouldn't have a hypothesis. The design of qualitative and quantitative studies differ. So in quantitative studies, there would be experiments or surveys, sometimes observations, whereas in qualitative methodologies, we use one-to-one interviews, focus groups, also observations, case studies. So these are things that I think might be familiar to you. A really big difference between quantitative and qualitative research is how we sample. So in quantitative studies, the aim is typically to obtain a representative sample. So we use things like maybe opportunity or convenience sampling, but the aim is to obtain a sample that represents the population of interest. So that population of interest might be, say, young people aged 18, or people who smoke, or people who use a particular NHS service. So that would be the population, and the aim would be to obtain a sample that's representative. By contrast, qualitative methods, the aim is to obtain a sample, and they tend to be much smaller samples. we use purposive or theoretical sampling, and the aim is to obtain a sample that maximizes diversity. So a good example would be, we might want to make sure that our 20 interviews that we conduct include women, people from different ethnic minority groups, people from different education backgrounds. So it might not be that if we recruited a representative sample and we only had 20 participants, we might not have that many, say, women or people from ethnic minorities if it was representative, but we are seeking their views. So the aim is to maximize diversity. And then research methodologies, rigor is really important to both, but how we assess rigor is different. So I expect that you have learned about reliability and validity and reproducibility in relation to quantitative methods. In qualitative studies, we're just as concerned about rigor, but we don't tend to use those same sorts of criteria. So we talk about other kinds of criteria. So, for example, credibility. So that's to what extent the interpretations that the researcher has made of the data are credible. What's important is that both of these methods rely on a systematic collection and analysis of data. So we tend to acknowledge that with quantitative methods, but sometimes it gets overlooked that qualitative methods also it's systematic data collection and analysis. So I thought what I would do is try and illustrate the sort of different methodologies by using a current example. So I want you to think about the vaccine that's been developed against COVID-19. So if we want to vaccinate the population against COVID-19, the first thing we have to do is develop a vaccine. So that's not part of psychology, that's biological sciences. But then we would want to establish the efficacy of that vaccine. So does it actually work or not? And we would do that by conducting a randomized control trial. So this is when half of the participants receive the vaccine and half receive a placebo, and you can look at the potential differences in outcome. So that's a randomized control trial, which is quantitative. But the next step, it's all very well having a vaccine, but even if we know that it works, we need to know whether it's acceptable to people. And this is what's been on the news a lot, hasn't it? That some people are quite hesitant about taking a vaccine. And this is where qualitative research comes into its own. So it'd be very difficult to understand why people are hesitant by using quantitative methods. And the reason it would be difficult, so we could send out a questionnaire that said something like, are you hesitant, and if so, why? And give a list of reasons for people to tick. But the problem with that is we as researchers might not be able to think of the reasons that people would give. So we can only think of the reasons we can think of. And a much better approach would be to use a qualitative approach, which is just to ask people in an open-ended way why they might not want to take the vaccine, and if there was anything or any way in which they could be persuaded. And that's where qualitative research is really useful. It's about understanding and exploring and explaining. This slide I put in, these are the different uses of qualitative methodologies in psychology, and I've put it in because I am a mixed methods researcher, so I use both quantitative and qualitative methods depending on what my question is. And we sometimes do a qualitative study because it's the right sort of approach for our research question, and it can be the right sort of approach when we want to investigate complex behaviours or processes. When we've got unexpected findings, it can help us understand them. But there are other sorts of use, things like instrument development, so questionnaire development, that I'm going to mention shortly. So there's a range of reasons we might use qualitative approaches. So I'm going to talk for a moment now about, in a bit more depth, about qualitative methodologies. And I think a useful way to think about them is in terms of their data collection and also their data analysis. So with data collection, there's lots of options available to us. We could collect data using one-to-one interviews, focus groups, which I think you've probably got a sense of, observation, ethnography, case studies. And then when it comes to analysing our data, I think you've learned a bit about content analysis. We tend to use something called thematic analysis, which is very similar to content analysis. So thematic analysis is looking at your data to identify patterns in your data. Discourse analysis, which is kind of the stories or the narratives that people will tell you about a particular phenomena. Conversation analysis, which is quite a specific technique, and it examines very precisely the sort of language that is used and the words that are spoken, and that helps us understand what's happening. There's something called IPA, or interpretive phenomenological analysis, which is when we ask people to talk about their experience in a lot of depth about a very specific phenomena. And then framework analysis, which is a way of handling lots of data from sort of open-ended responses, or where you've got a big qualitative data set. Framework analysis is useful. And then the one underneath is called grounded theory, and the reason it's there it's because it describes both the way data is collected and also the way data is analysed. So I thought maybe the best way to sort of illustrate qualitative psychology and what we do and how we do it and why we do it would be to describe some of the studies that I've been involved in the last few years. So first of all, I'd like you to meet Becca's at the top. She is a PhD student, or was when she conducted this work. Then I've got Kira, who is a neuroscientist, and that's Kira sitting in front of the data from an fMRI scanner. So she looks at what brains do. And then Shirley is a clinical psychologist. And this particular piece of work, this study was a one-to-one interview study. And the reason we conducted it, we were interested in how young people experience something called anhedonia. So this is a symptom of depression, and it describes the sort of inability to feel pleasure in things that somebody normally finds pleasurable. So it's called anhedonia, and it's an important symptom in depression. But the reason we wanted to conduct the study is it's only really, we know what it looks like in adults, but we're not very clear how it manifests itself or what it looks like in young people. So we conducted one-to-one interviews. They're called semi-structured interviews because you have sort of open-ended questions. So you just have a handful of open-ended questions, which are, can you tell me what that's like, or can you describe that? But there is some structure to it. We know that we've got a particular question that we want to answer. So we conducted these with 34 adolescents who were aged 13 to 18, and all of them had either a primary diagnosis of depression or they had elevated depressive symptoms. And we used, to analyse the data, we used thematic analysis, which is a bit like the sort of content analysis you're learning about. So what did we find? Well, we found, as I think we thought we might, that anhedonia in adolescents is quite different from anhedonia in adults. So for adolescents, it's not just about loss of interest and pleasure. It had things that we didn't expect to see or that we don't see in adults. And what was really important is it maps more closely onto the sort of negative symptoms that we see in schizophrenia, and this is novel to adolescents, and it's a bit similar to this the apathy that we see in Parkinson's disease. So the important thing is that it's not the same as it is in adults. And that's important because at the minute, the ways we have of assessing it were developed for adults and not for adolescents. So the measures that we're using to measure anhedonia in adolescents might not be quite right. And what that meant was that we were able to use the data that we collected in this study to create a pool of items that we then used to develop a new measure of anhedonia in adolescents. And if you look at the bottom of the screen there, you will see in little writing the citation for the paper that was published in a peer-reviewed journal that arose from this particular study. And I'm going to come back to that at the end. So as I mentioned there, Becca took another step. The next step in her research was to use this data we'd collected from the qualitative study to generate items, and then she went through what's quite a relatively complex process to turn that into a questionnaire. And then once she'd turned it into a questionnaire, there was another set of analysis to make sure that it was reliable and valid. And again, you'll see the citation for the paper at the end. So you can see, starting from that one-to-one qualitative study, we've learnt a lot. So now I'm going to talk about different sort of studies. So this was an observational study, and this was conducted by Stella, who was also a PhD student at the time, and my colleague Helen, whose research has been on an awful lot in the media recently, because she does research into the importance of adventurous play for children, and has been talking quite a lot about adventurous play that children have missed out on because of lockdown. So that's my colleague Helen Dodd. So Stella was interested in asking a question about children's food fussiness. So some of you listening will have a list of food that you don't really like. Some of you will describe yourselves as willing to try anything. Some of you will have been fussy eaters as children, others of you not. But food fussiness in childhood is extremely common, and it's a bit of a worry because the preferences that children develop in relation to food tend to be stable into adulthood. And we were interested in whether children's food fussiness, as it's reported by their mothers using this particular measure called the CEBQ food fussiness scale, does that match what actually happened? So Stella conducted a naturalistic participant observation. So this was a mum and a child, 67 dyads, and the children were given a lunch. And their lunch comprised both unfamiliar food and appetizing food. So it might be like a bowl of soup and some bread and a yogurt and some fruit. So some of that would be unfamiliar, some of it would be unappetizing, and similarly some would be familiar and appetizing. So it was structured in that way. And then we videoed the parents and the children to see what's happened, and we analysed that behaviour using a coding frame. So that's when you create a sort of list of behaviours that you're looking out for, and then you can analyse the videos, sort of ticking off those behaviours when you see them. What did Stella find? Well, she found that, in fact, mums are pretty good at knowing children's eating behaviour in terms of rejecting food and also accepting food. And they were sort of quite good at knowing whether children would consume familiar and appealing food. Other aspects of children's consumption, they were less good at predicting. But what's important from those findings is that this measure, the CEBQ-FF, that has been used really widely in the field by hundreds of researchers, instead, now we know that it maps onto children's behaviour. So you know that thing about reporting bias? So is what mothers tell us about their children, is that actually true? And from this study, we know that it is. And you'll see the citation for that work at the end there. And the last one I'm going to talk about is a focus group study. So you probably have got a sense of what focus groups are. I'm sure that some of you will have participated in focus groups or at least heard about them. And this was a study done by Lucas and Polly, who is a colleague who investigates child and adolescent, or particularly adolescent, mental health. And we were interested in particular in finding out what sorts of training secondary school teachers feel they need to enable them to support and educate their students around mental health. So this is about how your secondary school teachers talk to you about mental health and support young people with their mental health problems. So Lucas conducted 9 focus groups, each of which contained between 4 and 8 teachers. And in those focus groups, they spoke about these things, but we also asked them to look at the sort of training that was already available to them. And the data was analysed using thematic analysis. And what did Lucas find? Well, he found that teachers want training on how to provide and identify students who are struggling. Very importantly, teachers don't want to be therapists, so they want to support young people but have somewhere, someone qualified to send them to if needed. Training needs to be practical, interactive. They would like expert-led training. And that it's really important the resources can be adapted to particular schools or settings. So the outcome of that research was that a programme for training secondary school teachers is being designed, and importantly, it should address teachers' needs because it's being designed with that sort of focus group study underpinning it. And there you go. That's the citation for that work. So I know one of the things that we talk about, or that you're covering in your A-level, is research ethics, and this is important to all of the studies I've just described. So there's some things about research ethics that are common to all of the studies. So all of them were approved and allowed to proceed by the University of Reading Research Ethics Committee. So this is aligned with the BPS ethics requirements. There's also NHS ethics committees. So these are the sorts of committees that scrutinise whether what you're going to do is ethical. So you have to describe your research, you have to discuss potential ethical issues and how you might manage them. And it also includes, they look at the sort of information you're giving patients or participants and the consent forms to participate that they might use. It always includes information about how you're going to look after the data. So typically in qualitative research, data can't be anonymized. So we have to make sure that it's kept confidential and secure because it can't be anonymous. So in relation to the studies I've mentioned, that one-to-one interview study, well, if you think that was quite a sensitive topic for young people to talk about and for us to have data on, so we had to consider that as a particular issue for that study. The observation study, we had to think about what would happen if parents raised with us that they were concerned about their children's eating. So what would we do if parents raised that with us? That's an ethical consideration. And in the focus group study, we had to make sure that there were ground rules, things like, respecting other people and keeping what was said confidential and not interrupting and those kinds of things. So these were ethical considerations specific to each study. So the last thing I'm going to talk about is reporting and referencing and also peer review. So you will see this is the list of the papers that have arisen from the studies I was describing in my presentation. And all of them were reported using this very standardized format that we use in research. So we have an abstract, and that summarizes the research. We have an introduction, and that the purpose of that is to give the rationale for the study. So we consider other research and explain why this study needs to be done now. Then the methods section is like a recipe for the research. So you're describing in a lot of detail each step, and that will enable people to judge whether the study is of high quality or not. And in theory, you should give enough detail so that somebody else could follow your recipe and do that piece of research for themselves if they chose to. And that's the idea of reproducibility. And then the results section where we describe the findings, and a discussion section where they're discussed. And by that we mean the interpretation of the results, and also we consider the strengths and limitations of the research, and then their applicability. So why is this useful to know? So that might be it's useful for the next step in research, or it might be useful for say clinicians, or for teachers, or for other populations. Now you will notice that each of these papers that we've written, they're described here in a very specific way. So this is referencing, and you will always see the authors and the title, and then the journal that the paper has been published in. So for example, this first one was in European Child and Adolescent Psychiatry, and then very precisely which volume and the page numbers. And the reason we do that is to ensure that people can find our work easily. And so then in our background to a paper, if we make a claim and say Watson et al. Said this, it's easy to find the study where Watson and et al. Said it and make a judgment about whether that's a fair representation. So referencing is very important. And then finally, all of these studies, we drafted them, and then we sent our draft to the editor of journals, and the journal editor sends it to reviewers. So maybe 3 or 4 reviewers, and those reviewers are experts in the field, and then they scrutinize our work to establish that it was rigorous, that our interpretation of the data is reasonable. And this is really important because it's what maintains standards in scientific research. So this is how we differ from just saying what we want and saying we think this is true. This is a sort of quality check. So other experts in the field have looked at this and have said, yes, this is reasonable to say. And I thought that I would just finish with an example of what that looked like. So this is what Becker's paper looks like in the European Child and Adolescent Psychiatry Journal. And you can see the abstracts there, goes on to the introduction, lots of detail about being able to access it. And if you do go on and do psychology at undergraduate level, this is what you will be working with. This is where you will find out your information in sort of these kinds of journal articles. So I hope that has told you a little bit more about qualitative methods, a bit more depth, and in particular given you a flavour of how they are used to conduct real life research that's being used by policymakers and by clinicians and by other stakeholder groups, and sort of giving you a bit of a sense of what studying psychology and qualitative methods in psychology at degree level might be like. So thank you very much for listening.
Thank you, Kate. If I can get my camera on. And I think Kate has joined us now, and hopefully Sam is still with us. We can just maybe answer a couple of questions. One of the questions I think that would be useful to think about is when people are trying to choose a psychology course. I mean, there's lots of courses out there for psychology. It's a very popular degree. What kind of advice would you give them when they're looking at courses to help them choose the right one for them? I don't know who wants to go first. Shall we come to you, Kate?
Oh, that's a really good question. I think one of the first things I would suggest students or applicants look really closely at is whether the course is accredited or not. So there are BPS accredited courses, but there are some courses that aren't, or there are some courses where you have to do certain modules for it to be accredited. And that becomes very important for students who want to go on and pursue a career in psychology. So typically, if they want to go on and be a clinical psychologist or an educational psychologist, they will need an accredited course. So that's really important. And the other thing I would say is that school psychology is such a broad discipline that schools can really differ in the sort of everybody offers the same core components if it's accredited. So most of it's very similar, but the sort of flavour of it will vary according to different schools. So, for example, at Reading, we've got lots of people who are neuroscientists and clinical psychologists and health psychologists. So the sorts of modules that we tend to offer and the projects we tend to offer kind of look a bit like that. So I would suggest having a good look at the modules and seeing what sorts of things are offered in quite a lot of detail.
Okay, great. Sam, anything to add from yourself?
I agree with all of those points. I think accreditation is important depending on what you want to do. If you're planning to have a career in psychology in some way, then yeah, accreditation is really important. Some people aren't. Some people want a psychology degree for other reasons, and then it's actually, there's more flexibility in some courses because they don't have to stick to these core things. But I think if you want to be a psychologist, then looking for accreditation is an important thing. And yeah, looking in detail at the specific courses, what do you want to do with your degree? What specific parts of psychology are you interested in? Because it's such a broad subject. Look at the staff there, look at the kind of modules that are offered, exactly what Kate said, and see which of those interest you. And look at things like, do you want to do a placement year? Does that university offer a course with a placement year? Do they have joint honours things where you can do psychology and criminology, for example? So look at specific stuff like that.
Yeah, and I suppose the important thing for people to realise that's different to when they're at school or college is there isn't a national curriculum for universities, and a lot of the teaching, well, all the teaching is informed by the academics who are teaching their research interests. So you will get a wide variety of modules. And I've got a question for you, Sam, although it comes from something that Kate mentioned in her talk. She talked about PhDs, and I'm aware you relatively recently completed a PhD. So would you like to explain what a PhD actually is?
Yeah, how long have you got? I finished mine maybe 2 to 3 years ago now, and I now have a PhD student of my own. And actually, it's been really interesting because a PhD is very different depending on where you're doing it and what you're interested in. So for me, a PhD was a bit like a job. So I did my undergraduate degree and a master's, and then I was in a lab working, doing what was basically a job, just not a very well paid one. Whereas my current student now, he's more like a student. He's not attending classes, but he's very much sort of working on his own, doing research on his own. And it varies a lot, but it is very different from what an undergraduate course would be. So there's not really classes usually, or there's not many classes. You spend your time researching on a very specific question, and then you basically write a book about it at the end. I don't know if you'd agree with that characterization, Kate. Is that?
Yeah, I think that's pretty fair. I think it's a research training. In psychology, it's a research training. So you have the opportunity to work on typically a program of research. It takes about 4 years. You work very independently, usually with just 1 or 2 supervisors, and end up knowing more and more about less and less, usually.
That's a very good way of describing it. One of the things that many students might be a little bit concerned with is that they haven't managed to complete their whole A-level class. They might feel they've missed out on some things. When they go to university, is that something that should concern them? Yeah,
I mean, I think, I don't know about Sam, but this is something that at Reading we're really preparing for. We're very aware that students this year will come, you know, having had a somewhat different experience. We're trying to think about that, not just the idea that they might have missed things, but actually they've potentially really managed to develop some sort of qualities and skills that previous years haven't. So in terms of flexibility and resilience. So we are definitely preparing our courses and especially that first term with that in mind, but bearing in mind that most, certainly a big proportion of students who do psychology don't do psychology at A level. So I don't think students should be concerned about not having that content knowledge. And I think that probably the skills that you need to undertake a degree, you will have developed over most of your secondary school life. So I don't think students should be concerned about arriving without the same skills. And you know, we're kind of ready to catch people up if that's needed.
Brilliant. Anything else to add, Sam?
I mean, yeah, I'd say at Staffordshire we're very, very used to having. We have people, for example, who have been out of education for quite a while and are coming back to it, and we have lots of people who don't have any A levels and they've got other qualifications instead. And I think it's very normal for most, I'd say all psychology degrees. The first term or the first year is very much spent getting everybody up to kind of the same level, right? So some students get really a bit bored because they're not necessarily being entirely challenged in the first year, but it means that by the end of that, everybody is hopefully at roughly the same place, and then you can start on with the trickier stuff after that. So we put a lot of work into getting everybody up to the same level. So I wouldn't worry about that too much. That's what I'd say.
Great. And I'll ask the question: what do you think makes a good psychology student?
You go first, Sam.
I think just putting the time in, being willing to accept that what you need to do to be a good student at university is to do the learning yourself, right? It's you spend sort of school A levels very much being given stuff to learn, and the best students, psychology or pretty much anything you do at uni, you're going to do better if you are willing to put the time in, put the effort in, and direct your own learning. Ask for help with that if you're not sure where to look, but being able to sort of do that and go out and look for the information yourself, find what you're interested in, do the research independently. That's what makes the best students, I think.
Yeah, definitely. I'd really second that. I think particularly, probably I didn't really know what A levels really looked like until I had a daughter doing A levels, and then I began to realize why sometimes when students arrive at university it's a bit of a shock. And one of the things I'd say is that there's not that same sort of if you're asked to write an essay, students can get a bit sort of frustrated because they want to know what they should put in their essay. And at university, I would say, well, you know, what do you think you should put in your essay? So it's sort of much more independent learning, but that's great. It means you can go in directions that, you know, that interest you more. But certainly knowing what psychology is as well, knowing that it's both numerate and literate, that you're going to need to be able to write essays, but also there is that component to psychology that involves data analysis. The maths isn't hard, statistics is not like maths A level, but it does have that numerate element to it. Yeah, so I think kind of really knowing what you're going into.
Okay, and I'll ask you both a final question. What's the one reason you would give for someone to go and study psychology over and above other subjects?
Kate?
Well, I would say that probably, to me, I did my psychology degree a very long time ago. I think that the breadth of the discipline, there are so many different bits of psychology. I can't imagine anybody not finding part of it of interest to them. So whether you're interested in how children develop language or neuroscience, how brains work, you know, and scanners, or the sort of my field of psychology, which is health, you kind of living and breathing psychology all the time. So when you go and study it, it's really, I think, really easy to apply to yourself and the world around you.
Yeah, I think mine's kind of related to that, actually. It's because it's so broad, the skills that you come away with a psychology degree actually there's so many, because you're doing essay writing and things, so the kind of things that you get with some more humanities-y subjects. But also, like Kate said, there's data analysis, right? You learn to take some numbers and turn that into something meaningful. And you've also got qualitative stuff where you're able to analyze textual information, like Kate was talking about. So you come away with probably a broader range of skills than you would with most other degrees. I think that's what I'd say.
Yeah.
Degree.
Yeah.
Well, I must admit I'm biased because I did a joint degree in psychology and sociology. So psychology is a subject that's quite close to my heart. So it just remains for me to thank Sam and thank Kate for this afternoon. And hopefully we won't be here next week, but we are returning the week after to look at dance. So I look forward to